In my last video, we covered the absolute units known as giant viruses, a collection of viruses so incomprehensibly large they were initially mistaken for bacteria. In this video, we're going to go to the opposite end of the spectrum. Instead of the largest viruses, we're starting with the smallest true viruses and going smaller and smaller until we reach the smallest virus-like entities we can find. Collectively, these things are called subviral infectious agents, but for the sake of this video, I only want to look at the agents that still contain a whisper of life. It'll make sense when we get there, trust me. What creeped me out about giant viruses is how uncannily similar they operate to cellular life. What freaks me out about subviral infectious agents is how they collectively feel corrupted. Corrupted like my edgy OC. Corrupted like data is corrupted. To draw a physics analogy, when we're talking about viruses and bacteria and yeast, I think of Newtonian physics. Push a block and it moves. Drop a ball and it falls. The rules make intuitive sense. But once you start slashing away features of pathogens, you reach another dark valley of reality. One where individual molecules themselves can still resemble some kind of degenerate version of life. Let's start with the porcine circovirus, the smallest, most simple true virus I could find. The porcine circovirus type 4 consists of just 2 genes, one gene that contains the instructions to make the capsid, or viral protein shell, and one gene that helps replicate its own circular stranded DNA genome. This really is the minimum number of genes a virus can have. This virus's entire shell is just repeating units of the same protein. Compare that to a medium-sized virus like adenovirus, and you can really get a sense for how pared down this virus actually is. There are other viruses in the porcine circovirus family, all of which share a similar genome structure. Some of these viruses have genes crammed into other genes, a very efficient way of packing a lot of useful information in a limited number of nucleotides. All of these viruses are capable of infection, but not all of them are capable of causing disease. And that got me wondering, what kind of disease could you actually have with such a simple structure? One of the diseases this virus causes is simply called porcine circovirus associated disease. Real clever, I know. This virus is extremely widespread amongst commercial flocks of pigs and can cause a general degradation in body condition, infecting lymphocytes, weakening the immune system, and opening the door to other infections. The most noteworthy of this family of viruses is PCV2. This virus has 11 genes crammed into its hyper-efficient genome, a bunch of which are doing things you would expect a virus to do: evade, attack, and destroy the immune system. But the crazy thing to me is, PCV4, the version of this virus with just 2 genes, is also capable of causing disease. This virus was discovered not too long ago. The mechanism by which it causes disease is still unknown. Maybe there are cryptic genes hiding even deeper within its genome. Maybe this simple protein code is doubling as an immune system killer. Maybe the DNA or the RNA that it produces has some kind of function, or can give rise to multiple products. Who knows what the future of this virus holds? Maybe one of you will crack this code. Next up, we have the satellite virus. This entry on the list is technically a functional virus, given the circumstances, but it's just missing a few things. Satellites devolved a dependency on other viruses. Maybe they're missing a component or two from their viral replication machinery. Maybe they're missing a component of their capsid. This dependent relationship with other viruses may or may not stifle the fitness of their helper virus. A satellite virus that infects a giant virus is given a special name, called a virophage. One of the most famous virophages around is the Sputnik virophage, named after the fact that they orbit larger viruses. The Sputnik virophage parasitizes the mama virus, close relatives of the Mimi virus I talked about in my video about giant viruses. Sputnik alone does not contain all of the necessary instructions needed for its own viral replication. It relies on mama virus for crucial replication machinery. Evolutionarily, this makes sense. Why code for your own machinery if you could just steal someone else's? This is kind of the mantra for any parasite. In a sort of way, viruses having their own biology hijacked by other viruses is a form of cosmic karma. While not all satellite viruses produce a penalty for their host, Sputnik co-infection was found to have decreased mama virus replication by 70% and reduces the lysis of their host, the amoeba, by 3-fold. The enemy of my enemy is my friend. Satellite viruses don't just cause trouble for their helper viruses, they're also disruptive for human health. You've probably heard of viral hepatitis, a disease in which the liver is infected with at least one of several different viruses listed here. Except one of these viruses is a teensy bit less than a full virus. Hepatitis D is a satellite virus that relies on co-infection with its more well-known cousin, hepatitis B. But this satellite's a good bit weirder than Sputnik in my opinion. Hepatitis D doesn't code for its own envelope. Its replicating genome just appropriates Hep B's viral code, cosplaying as a completely different virus. It kind of screws my conception of what a virus identity even is. Since the coat is from a different virus, is Hep D just the genome inside? It's really weird to think about. So weird that depending on who you ask, Hep D might be better classified as a satellite nucleic acid instead. Hep D co-infection with Hep B is known as the most severe type of hepatitis infection. Hep D might not be a full virus on its own, but what it can contribute to pathology is the singular protein its genome encodes for. Let me repeat that. This satellite virus codes for one protein. This protein, called delta antigen, comes in two different flavors, a small form and a large form, caused by host enzymes replacing the stop codon on its RNA, extending the protein sequence by 19 amino acids. These two forms of the same protein are called isomers, and surprisingly enough, they have distinctly different functions for Hep D. But the reason why the host ultimately suffers for this is because this delta antigen is also immunogenic, meaning that when T cells, members of the adaptive immune system that can learn, recognize, and destroy pathogens, encounter this protein, they freak out and go on a rampage, destroying liver cells in their quest to vanquish this virus. Not only is the antigen immunogenic, it's also cytotoxic in its own right, a deadly combination for host cells. Remember, satellites are almost autonomously functioning viruses. The same cannot be said for our next category. By chopping off even more functionality, we get to our next category that I'm calling the glitches, starting with a literal defect, the defective interfering particle. These entities arise from mistakes made during the production of a virus. Because biology is just weird shapes bumping into one another, accidents at a molecular level can occur during replication. Polymerase, the class of enzymes responsible for nucleic acid replication, can just slip, and whoopsie daisies, a whole segment of the viral genome is just erased. What arises from faulty replication is a defective interfering genome, which, when it gets packed into a viral protein coat, becomes a defective interfering particle. Named as such because with a mangled genome, this thing can no longer be called a productive virus. It's broken. The first DIPs were discovered in influenza cultures back in the 1950s. I made a whole video on influenza, and in that video I made the claim that influenza's blazing fast replication speed and propensity for mutation makes it a disease control nightmare. Because influenza replicates so quickly, any mutations that would result in the production of a defective virus would be drowned out by the billions of copies of properly replicated viruses. But this turns out not to be totally accurate. On a species level, this is true. Influenza's replication rate can deal with its high mutation rate, but influenza isn't totally unaffected. These are defective interfering particles. They can accumulate and they can hamper influenza's ability to infect cells. Because their genomes have been shortened, they replicate much faster than the full-length virus. Hey, editing Phi here. DIPs do replicate faster than their correctly formed cousins, but attributing their replication rate to shorter genome size is an outdated concept. Faster replication is more likely to do with the shorter genomes accidentally generating stronger promoters, which makes them tastier targets for polymerases. And by replicating faster, they compete for protein shells and slow the ability for real viruses to be made. You would think that DIPs are another form of karmic justice. That evolving the high-risk, high-reward lifestyle of fast replication and mutation must surely come with some drawback. Surely. But this isn't necessarily the case either. DIPs are quite inconsistent and difficult to study. It's actually not entirely clear what DIPs do in naturally occurring infections. It has been observed that especially lethal strains of certain viruses appear to have mutated in a way that decreased the production of these DIPs. Which makes a lot of sense. Fewer defective viruses mean a nastier, deadlier infection. But remember, the goal of all self-replicating entities is to persist, not to kill. This isn't Plague Inc. You don't win by killing off all of your hosts. DIP production is related to viral infections that persist, causing prolonged misery for the host, and continued survival for the virus. acting as a kind of tuning system for infection strength, a mediating thumb on the scale of homeostasis. If I had to guess, if DIPs were truly harmful for viral fitness, viruses wouldn't be making them in the first place. It makes sense to me that these glitch viruses have stayed in the through line of evolutionary history for their ability to ensure the continued existence of viruses. Bizarrely weird guardians of both viral survival and host survival. The next glitch entry is the Viriform. The Viriform is an endogenized viral element, meaning it is a virus that snuck its genome into the host chromosome. This is a pretty general concept and applies to a lot of virus biology. Most famously, human immunodeficiency virus, or HIV, is capable of inserting its own genome into the host cell, forcing that cell to create copies of HIV for the rest of that cell's life. But what if the hierarchy was flipped? What if instead of the virus hijacking the cell hijacked the virus? Viriforms happen when an endogenized virus has been co-opted by its host for an important function in the host's life cycle. This kind of thing happens so rarely that the Viriform category was created just to make sense of the existence of one virus family in particular, the polydnaviruses. This group of viruses infects parasitic wasps and forms a mutually symbiotic relationship with them. This means that polydnaviruses get to permanently hang out in wasp cells, meaning as long as these parasitic wasps continue to survive, polydnavirus DNA will persist, completing the objective of life. Cells are generally not too fond of invading viruses, so in order for the penalty of virus infection to be offset, there must be some kind of trade-off. So what do polydnaviruses do for their wasp hosts? In exchange for permanent free room and board, the polydnaviruses will shut down the immune system of the caterpillar, the prey of their parasitic wasp hosts. Remember, Viriforms are endogenized viral elements. Evolutionary time has degraded their identity as a true virus into something that is virus-like. Within the wasp ovary cells, this degraded virus begins to produce viral particles that pack useful viral products into neat little packages. Because the Viriform itself is no longer a true virus, these packets are not true viruses, but simply containers carrying a paralyzing payload. Once the wasp injects its eggs and viral particles, the viral particles go to work suppressing the caterpillar immune system, enabling the wasp eggs to thrive within the caterpillar. Wasps that cannot make this Viriform fail to inject viable eggs into the caterpillar, as the caterpillar immune system will eliminate the threat. I need to take a breather here. Polydnaviruses are evidence of a host taming a virus, and how cool is that? I know it can be a tempting trap to personify biology, but this is a YouTube channel, so get off my back. And my purpose is partially to engage your imagination. I can't help but wonder about the experience of viriformation. Some could say that the Viriform is the idealized final form of a parasite, a true symbiote, a perfect merging of existence. An end to the eternal war between host and pathogen, a conclusion that so rarely ends with everlasting peace. But others could say that the Viriform is an abomination, an abandoned organism stripped of its former identity, glitched code that endlessly attempts to copy itself, failing for all of eternity, screaming into the void of oblivion, and yet because its failure is exploitable, it will never be allowed to rest. Endogenous viral elements have their fingerprints all over biology. Famously, the primate gene syncytin was a viral element that was co-opted to regulate placental development. But because the syncytin gene doesn't produce viral particles, it doesn't get to count as a Viriform. One quick shout out to genetic transfer agents, a type of Viriform specific to bacteria. Much like polydnaviruses, these viral agents are incorporated into the bacterial chromosome. But their viral particles are used to carry random bits and pieces of host DNA into whatever bacteria are infected next. Because genetic transfer agents aren't true viruses, the infected hosts don't make new viruses, but instead can incorporate DNA from other bacteria in a kind of peer-to-peer file sharing system, yet another mechanism by which horizontal gene transfer happens. Before we continue, I do have to acknowledge, yes, Prions are a type of subviral infectious particle. These special proteins take on a terrifying property when they're folded in the wrong way. They are capable of misfolding other prion proteins. Many of life's most horrifying diseases are propagated in this way. as piles and piles of misfolded hyperstable protein junk accumulates within neurons, killing them, leading to a slow, mostly inevitable decline. I won't go too deeply into prion science. I've already done a whole video on prions linked here. And not to toot my own horn, but I think it's pretty good, so go check it out. The reason why I put prions in the glitched biology category is that there's something very haunting about a bad protein corrupting other proteins, leading to an exponential accumulation of debilitating junk. While the prion is technically the smallest infectious agent, I don't put it last on this list for one simple reason. I want to keep this video about infectious agents that still have echoes of self-replicating life. And a prion, while it replicates via misfolding other proteins, it doesn't truly synthesize copies of itself. It simply causes other proteins to conform to its own will. We have finally reached the final category of subviral infectious agents. If viruses are the atoms of the living world, then these three agents are quarks, the absolute fundamental subdivisions of self-replicating life that we know of. But before we get into them, a super quick shout out to my YouTube membership. I believe all of my science content should be free on YouTube, but if you want my thoughts on non-science subjects or emojis to use during my educational live streams, please consider joining membership to keep this project going. Now, back to the final category of subviral infectious agents. The first agent we'll talk about in this segment is something I teased in my giant virus video. The obelisk. Named after the fact that this agent is nothing more than a single-stranded circular RNA molecule folded into an ominous rod shape. These agents were very recently discovered, their existence only becoming apparent last year. Since then, the biology world has been abuzz with curiosity surrounding these bizarre pieces of nucleic acids. So, what the heck even are obelisks? It's really hard to say. Their RNA genome is unlike any other organism we have sequences for, and again, from the giant virus video, this means that it is extremely tempting to put obelisks in their own special category of life. Obelisks do not code for their own viral coat, nor do they co-opt one from a helper virus. These are complete parasites that just require hosts for their own replication. They do encode for useful products, proteins called goblins. Wait, no, sorry. That's autocorrect. Proteins called oblins. Two oblins have been discovered so far. Oblin 1 and Oblin 2. This discovery is so hot off the presses that we have no idea what it is these proteins actually do. Our go-to tool for generating hypotheses based off of homology, that is, an existing evolutionary relationship with an organism we do know about, doesn't work here, because again, obelisks are freaky little aliens. So the best guess scientists have for these oblins is a structural hypothesis. Oblin 1 looks like a globular protein and maybe binds RNA. Oblin 2 looks like a leucine zipper protein that could potentially act as a kind of protein domain adapter. Some obelisks can make ribozymes, which are RNA molecules with catalytic activity, often cleaving replicating nucleic acids as a part of their own replication cycle, adding one more potential tool obelisks have to manipulate biology. There's kind of an issue with regards to finding these mysterious obelisks. They're not incredibly long, and since they don't appear to have an existing evolutionary relationship with other organisms, how do you know a piece of RNA is actually an obelisk? I'm not a smart enough cell to know the answer myself, but I did want to shout out this group for attempting to build an automated pipeline to find predicted obelisks, which they name the Tormentor. Oh my god, I just got that. Oh my god, you nerds. So what organism is parasitized by obelisks? It turns out, obelisks are everywhere. In the human microbiome, if you look for their faint signature, you can find them within Streptococcus sanguinis, a common resident of the human oral microbiome. Mammalian bacteria are not the only hosts for obelisks. They're also found in an environment we so rarely get to talk about on this channel, the ocean microbiome. Obelisks were found to be highly abundant in prokaryotes isolated from all temperate and tropical oceans in the world, as well as the Arctic samples isolated from the Terra expedition. And as of my current research, their impact on the biology of these systems are totally unknown. They're really weird to think about. Finally, we have viroids and virusoids, what I believe to be the absolute minimum units of infectivity. Okay, I'm gonna be real here. Virusoids were incredibly hard to place in this list. I chose to put them here because virusoids are kind of like a mishmash of other infectious agents we've already talked about so far. Virusoids are a specific type of single-stranded circular RNA satellite virus, very similar to the obelisk. Virusoids, unlike the obelisk, are encapsulated by proteins made by their helper virus. But unlike most satellite viruses, virusoids don't code for any protein. That's right, the hepatitis delta virus, coding for one singular protein, is still more complex than the virusoid, which codes for absolutely nothing. While virusoids don't code for protein, many of them still contain some kind of information that leads to the production of something catalytic. And that something is the ribozyme, that catalytic RNA molecule that obelisks also make, a potential throwback to the earliest days of life itself. Virusoids act as companion viruses to their helpers, exacerbating diseases that are especially relevant for commercial crops. How could a measly piece of RNA even cause disease? We're going to hold off on that line of questioning because the answer to this is also very similar to the final entity on this list called the viroid. Viroids are a lot like virusoids in that they are single-stranded circular RNA molecules that don't code for any protein, except unlike the virusoids, they don't even have a protein coat that they steal from helper viruses. Viroids are truly minimal units of self-replicating parasitism, floating molecules of RNA that can't code for protein, whose only function is to leech off of the host polymerase for successful replication. While most virusoids make something that is a functional ribozyme, half of viroids don't even make that. How bad could an RNA pathogen that doesn't even make protein even be? If you're an agriculturalist, these things could spell disaster. Viroids and virusoids are known plant pathogens. When a cell is loaded up with these pathogenic RNAs, these RNAs replicate, and after they're processed, either through their own ribozymes or host RNases, they form RNAs that can bind to host mRNA, which triggers their destruction through a mechanism called gene silencing. Remember, the central dogma states that information flows from DNA to mRNA to protein. Without mRNA, protein cannot be made. The direct result of this silencing is that important parts of the plant's protein production are simply shut down, and infected cells, as a result, become compromised. I'm about to wrap this video up, but please stick around for just a bit longer. I have an important channel announcement. On January 10th, starting at 9 AM EST, I'll be holding a 24-hour charity live stream to raise money for the Breast Cancer Research Foundation. I'll be talking about the science that you will directly fund, from basic mechanisms of breast cancer pathogenesis to research model development to drug design and more. Hours of research will be punctuated by more lighthearted segments featuring some very special guests. I started doing these charity marathons last year, and I can't wait to get back into the fray. We started this journey looking at viral titans, huge pathogens that dwarf some bacteria in size. And we will end with the smallest, most simple self-replicating pathogen, a humble strand of RNA whose only crime to the host cell is merely existing. Viroids bring to light something that I've always felt was amazing about biology. Self-replication imbues everything with a kind of lifelike property. Through natural selection and evolution, every self-replicating molecule is afforded a chance to survive. And only the luckiest, most tenacious molecules get to continue to be written about in the textbooks of biological history. Before we go to the credits, I need to acknowledge something about my giant virus video. I missed out on one particularly insane virus. Alpha pithoviruses are giant viruses that are disgustingly huge, bigger than even the Pandora virus. I missed this virus because, can you blame me? This thing's got a total of 10 entries on Google Scholar, a very underground virus that eclipses even the mighty E. coli in size. Thank you, commenters, who have pointed this out to me. Thank you to all my members who continue to support this project. You are all rock stars.